Hi everyone, welcome to this new Guénin Life Science webinar that will be dedicated to atopic dermatitis. We'll just wait one last minute for last people to join. After this brief introduction of who we are, I will now present you epidemiology of atopic dermatitis. Atopic dermatitis, also known as eczema and atopic eczema, is one of the most common inflammatory disorders, affecting up to 20% of children and up to 10% of adults in high-income countries. The causes of AD are complex and multifactorial, and AD is characterized by intense itching and recurrent eczematous lesions. AD has heterogeneous clinical presentation and different endotypes exist. Therefore, currently available therapies only help certain subpopulation of patients. Eczematous lesions also show age-related distribution, and you can see in the middle of the slide a red dot showing where most of the lesions are located depending on the age. Different results indicate also that environmental factors are of importance in the etiology of AD. We can find the so-called hygiene hypothesis and the gradient between urban and rural regions. Current treatments involve topical corticosteroid as a first line of treatment. We can find also UVB phototherapy. And now there are also monoclonal antibodies available, such as dupilumab, which targets IL-1 receptor subunit alpha. We can find also JAK inhibitors, such as abrocitinib and upadacitinib. The causes of AD are complex and multifactorial. And even if molecular mechanisms involved in this pathology are more and more understood, there are still some debates about the factor triggering its development, the so-called inside-out and outside-in paradigm. Inside-out hypothesis suggests that inflammations precedes epidermal barrier dysfunction, and we have shown that the inside-out theory was enough to induce AD in the humanized mouse model. On the contrary, the outside-in one suggests that impaired barrier function precedes AD development. This is most likely dependent on filaggrin mutation, which is found in 20 to 40% of patients. Regardless of the mechanism, AD development is mainly associated with Th2 cytokines, but also with Th17, Th22, and Th1 cytokines, and their combination determines the different endotypes. In this picture, we show stage-based pathogenesis of AD and the main mechanism of this pathology. Even if some stages occurring in AD are described, please note that this scheme only comprises one of the possible mechanisms for AD development. For each step, we will show you some assays that are available at Guénin to study this pathology and assess the effect of compounds. First, on the left, we can see that in non-lesional skins of AD patients, epidermal barrier deficiency is observed. This barrier deficiency is associated with a reduced diversity of the surface microbiome, characterized by an increase of Staphylococcus aureus proportion to the detriment of other microbial species. Several assays are available to study the effect of microbiome dysregulation, and particularly the impact of Staphylococcus aureus associated or not with filaggrin deficiency. On this slide, you can see the topical application of Staphylococcus aureus at the surface of epidermis will induce a decrease in filaggrin expression. You can see here a decrease of the red staining in the middle epidermis. This inhibition can be rescued by the application of compounds. See the epidermis at the bottom of the slide, where we can see an increase of filaggrin expression after a topical application of compound X, for example. Since filaggrin mutation can be found in nearly half of the patients suffering from AD, we can propose you to work with reconstructed epidermis in which filaggrin expression was altered. either by specific Th2 cytokines. You can see here this kind of epidermis, or by silencing the expression of filaggrin genes, either initially at seeding of the cell, so before the wall development of the reconstructed epidermis, or using inducible silencing that will allow to silence filaggrin gene expression at each step of epidermis development.